And did I do it right? Yes. Okay. So I'm sure there will be other people logging in. I just have not got a clue what happened. I noticed when I was broadcasting that I was frozen, couldn't see myself. I tried to chat on the chat line and not working. I went and couldn't even open or close any kind of PowerPointy stuff. My computer was just frozen up. So when I went to restart it, it acted like, I don't know, it had crashed or whatever, and it had to reboot and re-update, and it took forever. Well, it seemed like for me, it took forever. All I could think of was people sitting there at home waiting for me to send a new email out. So I went to a different computer that I can't really broadcast on, but I sent out an email saying, stay tuned. And so hopefully there are people who have not given up on me and are, taken off and gone swimming or whatever people do now during the virus. Anyway, I want to go back. I'm recording and everything now, so I want to go back. I don't want to waste any time. I swear it's updated the very way that the bottom of my computer looks. I wanted to show you something, and gosh, you know what? I want people to take a look at this text reading that's mentioned here on the agenda. And I have to figure out how to give you that. And so what I think I'm going to do is this. Okay, come on, go down. I'm going to go here, go back to the agenda. Okay. I mentioned a number of things here, but here's something that's important that I did not really mention, and I need to. And it has to do with a free chapter, a really pretty good chapter on tissues and histology that I would really like everybody to read. But since I don't post these daily agendas, I think what I'm going to do is I'm going to jump right down here to another PowerPoint, one that I'm absolutely going to talk about, okay? And since I'm sure there's some crazy good way to do this, but I don't know what it is. So I'm just going to put that link here because I absolutely expect I will post this document and you will be able to look at it. So here's this is the text that is on tissues that I'd like you to start reading as soon as you can. Should be pretty easy to open. It's going to take you to a page. You might have to put your email in order to read it, but it is really a pretty well written chapter on tissues. It's still loading, of course. And honestly, we don't need to look at it now. I just opened it to make sure that link worked, which that link did work. So for what it's worth, this is a chapter in a textbook, an open source textbook that we can use on tissues that is very good. And so in addition to everything else I'm asking you to do this week, because we've got a crazy and busy week, I'm asking you if you would start taking a look at this chapter and reading it. It's going to make things, if you start reading it ASAP, it's going to make things later in the week make more sense, okay? Just for the record. Now, because it's in the, because it is on the agenda, which I don't generally post, I have embedded it in the very first PowerPoint that we're going to use today, and it's the second slide in the very first PowerPoint, so you can take a look at it. Okay, so on today's agenda, I've got a list of PowerPoints. The first one, the one I am going to start with and do part of at least today, I'll leave the rest to you to look at, is this one that's called cell membrane transport. And again, the second slide in the cell membrane transport one, the The second slide in the cell membrane transport is the text chapter. Okay, what does this mean? Error. Okay, I'm waiting for that. Now look, I appreciate everybody being patient with me. I mean, this is actually, we've been kind of lucky. This is the first time that the computer kind of crashed right at the beginning of the class, and I had no intention of canceling the class after that. This is a very busy week, so I got to make as much progress on material as we can today. So you know, if something similar to this should happen again today, and there's a significant amount of class time left, then we are going to absolutely have to just restart. That's the only way to handle it, unfortunately. Okay, so agenda. I want to leave it up here for a second, just so anybody who wants to copy it down and look at it can look at it. Oh, wow. I guess I didn't save that one change. Sorry. It wasn't changed when my computer crashed. So we're going to say do Friday, July 31st. Okay, so that's when the osmosis at lab at home is due. Okay, I better save this. Yeah, when my computer kind of crashed before, I guess I must have lost some little, little changes that I had made. So I better remember to save those things. So as far as this goes, I think we've gone down all of this. I do want people to have an opportunity to look at it, take a picture with your phone, write it all down, whatever. But, so here's the agenda for today, and I've already talked about it, but, I mentioned there'll be a new quiz posted tomorrow. Before class, it'll be due 2 days later. Exam 2 is going to be posted on Thursday. I'm going to get that posted as early as I can. It's going to be due Saturday, August 1st. Okay. And then please, when you get a chance, take a look at this text reading on tissues. And if you don't want to write all this down, I understand that. Then I have posted this link in the very first PowerPoint that I'm going to show today. Okay? All right. Well, if anybody has any questions about that, of course, you know what to do. Just jump on in and ask. All right? Believe me, if I don't know the answer to the question, I'm not going to waste any time and just tell you, hey, don't know. Okay. Oh, good. You wrote it down. Thank you, Emily. I appreciate it. Now it's on the chat line if somebody wants to just cut and paste it somewhere. All right. So, look, let's not waste any time. Time has already been wasted by computer problems. And like I said, I have to sort of admit that every day that we meet, I think, oh, we are going to have computer problems. And most days we don't have computer problems. So what can I do? Ever since I started teaching virtually, I've thought probably I need to get a better computer, a bigger computer, a faster computer, I don't know, whatever, a new computer. And, but I have not done it. I'm still working on my same old Dell laptop, which isn't a bad computer, I don't think, but I don't know if it's completely up to the virtual class thing and doing that well. Anyway, this is Cytology 3, Cell Membrane Transport.1. I'm going to work through it. If I end up tweaking it, I will, I'll update it to Cell Membrane Transport.2. I absolutely will post this PowerPoint as soon as class is over today. All right? So is everybody with me? Here we go. The text reading on tissues, I deliberately put that there so you would have a copy of it because I am going to post this PowerPoint. Look, this is all about transport across membranes. And like I said, there's a lot of detail here. I'm not going to go through this entire PowerPoint. I'm going to go through part of this PowerPoint, and then I'm going to just let you finish it up, okay? But let's talk about transport across membranes. I mean, this is a huge deal for cells. They spend a lot of their energy on moving things in and out of cells across the cell membrane. So basically, there's different conditions inside and outside the cell, and they have to be maintained for homeostasis. There's a continuous exchange of ions, salts, sugars, and other molecules between the inside and the outside of the cell. These solutes are found both inside the cell and in the fluid and spaces around cells. So here's a document I stole out of one of Guyton's medical physiology textbooks that I really like. And look, let me tell you right away, of course, I'm not going to make you memorize any of these, but let me just explain what you're seeing here. Look, it says intracellular fluid. This is everything inside the cell. That line right there is the cell membrane. This is inside the cell, intracellular. This is right outside the cell, extracellular. And of course, here's the line here that is the selectively permeable membrane that determines whether things move in and out. But take a look. You don't need to know what a milliequivalent is. It's a way of expressing concentration, okay? So sodium, ion, 142 milliequivalents outside the cell, 10 milliequivalents inside the cell. There's 14 times more outside than inside. Potassium ion, 4 milliequivalents outside, 140 milliequivalents inside. That's 35 times more inside the cell than outside the cell. Calcium, 2.4 milliequivalents outside the cell, 0.0001 milliequivalents. I mean, there are thousands of times more calcium outside the cell than inside the cell. Magnesium is 1.2 milliequivalents outside and 58 milliequivalents inside. So it's like a 40 times more inside the cell than outside. Chloride, 103 milliequivalents outside, 4 milliequivalents inside. The point is that there are really big differences between the extracellular and intracellular environment. I mean, big differences in the concentration of important things like chlorides and bicarbonates and sulfates and glucose and amino acids and you know, all kinds of gas values, etc. The bottom line is that that all of these differences here, how do those differences happen? And the answer really has to do with this line right here. I mean, this line right here that separates the internal environment from the external environment, that is the cell membrane. And this is just no coincidence. This is the cell membrane actively working to maintain the homeostasis in the intracellular fluid, regardless of what it is in the extracellular fluid. So it's kind of like when you think about it, all of these amazing differences have got to be due to this line right here, which is the cell membrane. Again, these numbers are just to sort of paint a picture of how different the conditions are inside the cell or outside the cell. So let's talk about the types of transport across membranes. One of the most famous ones is diffusion, and we'll talk about it today. There's unaided diffusion and there's carrier-facilitated diffusion. Unaided means the diffusion just happens by itself, and carrier-facilitated diffusion means the cell actually has installed a special cell membrane protein to help diffusion happen. Osmosis, which is sometimes just considered a type of diffusion, is another type of no energy kind of transport. Now, active transport, endocytosis, receptor-mediated endocytosis, phagocytosis, and pidocytosis, these all are energy-consuming processes. So when I say passive transport, what I mean really is the cell does not need to put energy into this process. The universe provides the energy. Now that may sound strange, but there is a lot of energy out there in the universe, and there are some processes that are supported by that energy. And that includes three of passive transport called diffusion, osmosis, and carrier-facilitated diffusion. Now, all the other ones we talked about are what are called active transport, meaning that they require cellular energy, energy provided by ATP. And that includes things like active transport, receptor-mediated endocytosis, phagocytosis, pinocytosis, and exocytosis. So all of these names, these are all names that will be defined within the PowerPoint, okay? We're jumping right in. So it is kind of interesting that there's this energy out there, kinetic energy. It's provided by the universe, and it's responsible for this kind of random molecular movement. All molecules move in some fashion. When molecules move, they bump against each other like bumper cars, and they move away, and they bump into other molecules, and energy gets transferred from one molecule to another. This random molecular movement is usually called Brownian movement. And let's just, for fun, I think that it's worth seeing if we can't just real quickly find good old there it is. Let's see if we can see a little short video of Brownian motion. All right, and I suspect that, okay, let's see. All right. Well, sorry. Should have just did this right.
In this lesson, we're going to have a look at the idea of Brownian motion for this, supported by a fluid. In this case, it's a bit like a game of snooker under the microscope. It's a challenge for you. Thinking about what we know already. Molecules. Each water molecule is too small to see.
I mean, there was a really cool Brownian motion video, which just has disappeared. It's similar to this.
Okay, so here's our last slide, and you can see the dimple on, and there. But let's increase the magnification a bit more so you actually see some Brownian motion. And there we go. So I adjust my magnification. I can see my little drops in there all jiggling around. And that jiggling around that you see is Brownian motion. So now your job is to pick a good candidate feed to trace its motion and measure how far it travels and given lengths of time. And to do the
Well, anyway, Brownian motion is a kind of motion that describes the random molecular motion of the universe, the energy that is provided by the kinetic energy of the universe. Really, things are in constant motion. The universe is in motion at the atomic level, as you know, and that makes things move around. Now, a concentration gradient is really an unequal distribution of molecules between two areas. So if you have a lot of molecules in one area and few in another area, then you say, oh, we've got a concentration gradient. High concentration over here and a low concentration over here. So one thing that can happen by diffusion is just the movement of molecules from being concentrated somewhere to being less concentrated and spread out in roughly an even way across. So the first kind of passive transport I want to talk about is a super important one called diffusion. Diffusion is the movement of molecules from an area where they're more concentrated and an area where they're less concentrated. The energy for this is supplied by the random molecular movement of kinetic energy. Um, Honestly, this is such an important process that a law has been created called the law of diffusion. And the law of diffusion says this basically. Let me stick it in here. Right? The law of diffusion. Yeah, molecules, it's a law, so. And this is what the law of diffusion basically says. Molecules, substances will move from where they are more concentrated. Well, did I lose an entire line there? The law of diffusion. All right. So I'm going to go back to the slideshow, of course, from the current slide. So here's what it looks like. Here's a bunch of highly concentrated stuff. And here it is very low concentration. And the law of diffusion says that when you have a bunch of stuff concentrated like this, over time, it's going to move to where it's less concentrated. If this happened for real, I would expect that what would happen is you have a bunch of this stuff, whatever it is, could be dye, it could be sodium chloride, you have a bunch of it somewhere. And over time, it spreads out basically evenly throughout this entire area. It moves from high concentration to low concentration. Of course, the little blobs are the solute, the dissolved stuff. The blue here is the solvent, which is water in biological systems. So diffusion can happen with a membrane present or with not, right? And so most of the time, though, when we're thinking about biological systems, we're thinking about diffusion happening where there are membranes set up to allow things to happen. Oops, what happened there? Right. So we've already talked about membranes being selectively permeable. They allow certain things across and they restrict or prevent other things. So passive transport is diffusion across a membrane in this case. We usually think of diffusion as just kind of any kind of movement of particles from one place to another, from an area of high concentration. Right? Now, when we've got membranes involved, that's a big deal. But, when I think of diffusion, I have to tell you, I think of some examples. Like when I was in a classroom, I haven't done this for years, but I used to get one of those cans of that really stinky floral potpourri, and I would spray it over in one little corner of the classroom, and I would ask people to tell me when they'd smell it. And I would spray a big clump of it, maybe 5 seconds worth of spraying, and then I would walk very carefully and gently away so as not to drag the scent. And I would ask people in the classroom, hey, tell me when you smell it. And, what would happen is in about 30 seconds, people pretty close would smell it. And in a minute, people would smell it further deeper in the class. Eventually, and I mean, it would generally only take a minute or two, it would appear everywhere in the class. The particles of that stinky potpourri from that can went from being highly concentrated in one little corner of the room to spreading throughout the entire room, and probably being roughly evenly concentrated everywhere. Okay? So, when we think about diffusion, we're thinking about these movement of these particles. Now, when we're thinking about biological system, we're thinking about diffusion across a membrane, and it diffusing from where it's more concentrated, and that could be inside the cell or outside the cell, to where it is less concentrated, which could also be inside or outside of the cell. So diffusion, the very first one we're talking about, diffusion is that kind of thing. Now, you know, when I was a kid, I had to get up really early and go to work, and every day I would basically say to myself, look, when the weekend comes, I'm going to sleep as late as I want. This getting up at 5:30 in the morning is ridiculous. I'm going to sleep as late as I want. So on Saturday morning, here it come, and I'd be laying in bed, and it'd be, you know, like 7:30 in the morning, and I'd be wide awake. And I would be laying in bed trying to pretend I was going to sleep the rest of the day. And suddenly, I would smell something, and it would make me want to get out of bed. And what I would smell was bacon. On the weekends, my mother would make bacon for breakfast. But here's the thing, that kitchen was way at the other end of the house. And yet those bacon molecules had managed to, where they were highly concentrated over that cast iron pan where my mother was making the bacon. But then they diffused out of the kitchen. They diffused across the living room. They diffused up the stairs, down the hall, underneath a little crack in my bedroom door, you know, a ½ inch wide. And then they started moving through my bedroom until one of them came close enough to my face, and I inhaled it. And my brain went, ooh, what is that? Uh-oh, bacon. Oh my gosh. That bacon had worked its way from the kitchen, out of the kitchen, across the living room, up the stairs, down the hall, under that crack in my door, all the way across, under the covers. Now it's wedged in my nose. When we smell things, it's because the molecules that make those things are wedged in receptor sites in our nose. And I used to think at that time, I better get up or I'm not going to get any bacon. The bottom line is that kind of process, where the smell one is a good one, where you smell something that happens way in the other end of the house, odds are very high that is just because of plain old diffusion. One of the processes that we need to think about when we think about membrane transport processes. You know, when I walk in the door at night, no matter where I've been or how long I've been gone, I immediately know if I have litter boxes that must be changed right away. The stench immediately tells me, uh-oh, there is a litter box somewhere with something very odoriferous in it, and the molecules from that odoriferous substance have managed to diffuse throughout the house, or at least to the front door where I can smell them. So, you know, diffusion is a normal part of our life and our thinking. The last comment down here, where it says small nonpolar molecules such as oxygen gas and carbon dioxide gas diffuse easily across the phospholipid bilayer of a membrane. Those gases like that will diffuse easily. If there's more oxygen inside of a cell than outside of the cell, then we will see oxygen diffusing out of the cell. If oxygen is more common inside the cell, then it is going to diffuse out of the cell. So again, you know, here we've got a cell membrane, a high concentration of something, 0 here. Slowly but surely it diffuses out until there's roughly an equal number of molecules. Molecules, like it says there at the bottom page, will move from their higher concentration to their lower concentration. That is certainly another way to restate the law of diffusion. For what it's worth, it was me, Steve Mackey, who created the law of diffusion. And I've for years tried to call it Mackey's law of diffusion, but nobody will allow that to happen. People just won't. They insist on just calling it the law of diffusion. So what about permeability? This is whether or not substances can cross the membrane. And what does it depend on? It depends on their size. If it's too big, it just won't fit through. Membranes have, size limitation. Whether it's hydrophilic or hydrophobic. I mean, the membrane inside is hydrophobic, remember, because it has stacks of lipid molecules. It's the phospholipid molecule that has at one end that is fatty fats and one end that is non-fatty and is hydrophilic. So anyway, whether things can move through membranes depends a little whether they're hydrophobic or hydrophilic, of course. Whether they move depends on whether there's a higher concentration on one side of the membrane than another. If there's the same concentration, then that doesn't mean they're not going to move at all. It just means that they are pretty much, you won't really be able to notice a big change. So, in this case, here's a nice phospholipid bilayer. And notice these non-polar hydrophobic solutes just kind of move back and forth through the membrane pretty easily. But then, if you are polar or ionized or you're solute, well, then technically you're supposed to enter the cell in a cell membrane. So there you go. Now, anyway, this is, let's say, extracellular fluid, and this is intracellular fluid. So remember, the two types of passive transport were simple diffusion, where a molecule simply goes right through the membrane, carrier-facilitated diffusion, where there is a protein carrier that's embedded in the membrane and goes all the way through to help. It has a pore, meaning a channel through it for the passage of molecules. It can be pretty general. It can allow a molecule of a certain size or shape to enter, and it doesn't really care. Or it can be really selective and go, no, I'm only letting glucose through. That's the only thing I'm going to be letting through. Again, this type of passive transport diffusion, it doesn't use any energy. Where did the energy come from? It's powered by concentration gradients. It's powered by, I know this is going to make me sound airy-fairy, but it's like the universe powers it. There's lots of energy out there, and so some of that energy can be used to power a process like this. In passive transport, which way would molecules always move relative to their concentration gradient? Well, what I'm asking here is that if you can see that there's more molecules in one place than another, what direction are they going to move if they're just doing plain old passive transport? And the answer is they're going to move from where they're more concentrated to where they're less concentrated. So out here, they show an image. Here's, a phospholipid bilayer. Here's the extracellular fluid. Here's the intracellular fluid. Here's a big blobby protein that happens to have a channel that moves molecules in of a certain size. Now, honestly, if all of these represent a solute molecule, then you kind of need to know what solute molecule it is to know whether how it would behave like this. But let's just say it was a gas like carbon dioxide or oxygen. We already know will pass through membranes pretty easily. Okay, now, before I leave this
question.
Yes, go ahead.
What determines whether or not the diffusion is going to be simple or carrier facilitated?
It depends really on whether the substance can diffuse. Well, here's what it sort of depends on. Is the cell highly motivated to move this substance? And does it move just by simple diffusion, right? If it moves by simple diffusion and it goes right through the membrane and the cell needs it, well, then the cell will be able to acquire it that way. But if, in fact, the cell needs a molecule, and a perfect example of this is glucose. And that glucose concentration is higher on the outside of the cell than inside the cell. But the glucose molecule is just too darn big, then it may absolutely require the cell to build a protein carrier. So, a
got
cell
you.
will build a protein carrier if it's necessary for the cell. Like one really important molecule that diffuses pretty well across cell membranes is water. And that seems a little odd just because water is pretty, the lipid center of a membrane is pretty hydrophobic. So you kind of think, well, wait a minute, how exactly can water get across a hydrophobic membrane? And that really bothered people quite a bit because the bottom line is that water does move perfectly through a membrane, right? And how it does that is, well, it could be how it moves across the membrane was not well understood until they discovered a specific protein for water called an aquaporin that is specifically designed just for water. Anyway, I hope that answered your question, sort of.
Okay, so it depends on just like the size, not the, or like stuff like that, but not the type of molecule.
Well, it absolutely can depend a little bit on the type of molecule. Like, is it big? Is it hydrophobic? Is it hydrophilic? Okay? So it absolutely could depend on that. But the bottom line is that it also can depend on the size, the shape, all of those kinds.
Okay, so it's just a lot of factors.
Yeah, a lot of factors. So I'd like to paint a picture. Hold on, I just want to go here for a second. I'd like to paint a picture here that of some of this. And so what I'm going to actually do here, is I'd like to post, there's a document I'd like to be able to show you, and I need to just grab it and post it in here. Okay, it won't take me very long to do it. Whoop. Sorry, I lost my mind there for a minute. Okay, now where am I here? And again, I apologize, that's me just sort of losing my mind. I wanted to put this up. All right, so that'll be there, and then I want to, a couple slides I'd like to put up here. Just give me 1 second, sorry. It will just take me a second to process this. Where is this? This is something that I just saw 2 seconds ago. It is absolutely here. I know it is. Okay, sorry. I'm going to go back here to this document, and I'm just going to resize it a little bit so I can show it to you. Okay. All right, so slideshows. Sorry, folks, I apologize. So here's just some notes I just want to post. I want these to be in the slideshow. Diffusion, physical process in which substances, atoms, molecules move from where they're more concentrated to where they're less concentrated. Like I said before, the universe is in constant motion at the atomic level. You know what I mean. Electrons are moving at 2,000 meters per second. I mean, there's motion at the atomic level, and that translates into energy, motion energy that sort of powers molecular movement with the energy provided by the universe, as crazy as that sounds. And then there's Mackey's law of diffusion that says substances will move from where they are more concentrated to where they're less concentrated if they can. Now look, can everything diffuse? No. You can prevent diffusion from happening. You can have one of those spray cans full of stinky potpourri and put your nose right to it and sniff it, but you won't smell it, because that metal can prevents diffusion from happening. But here are some observable patterns in diffusion behavior that I'd like to mention. Big molecules diffuse slower than small molecules. Right? The bigger a molecule is, the more energy it takes to move it around a little bit. And even though there's a lot of this kinetic energy, this universal energy that creates something like Brownian motion, the bigger something is, the harder it is to move. I mean, it has something to do with energy. It's just kind of like it's harder, it takes more energy to move a couch than it does to move a book, right? Now, here's another thing. As temperature increases, the diffusion rate increases. Now, actually, some people are going to say, well, that's totally logical, Steve, because temperature measures energy. And the more energy is available, the faster diffusion will happen. Molecules will bounce against each other more, bounce further away from each other, etc. Now, in cell membranes, though, there are some things that diffuse pretty fast and some things that don't. Gases, like oxygen and carbon dioxide, diffuse easily and fast. They do. They're nonpolar gases. They wiggle through cell membrane very easily, no channel or anything required. Ions, oh, and I mentioned water too. Water moves back and forth through membranes very easily because water is such an important molecule. And the only way it can move back and forth easily like this is through a channel called an aquaporin. That's what it says right there. Sorry if it's not as readable as it could be. I'm going to put it in the chat line. The word is aquaporin. Okay, back to here. So, water, gases diffuse easily and fast. Ions, like sodium, potassium, and chloride. I mean, these are all important ions. We see that they have, concentrations in the intra and extracellular fluid, but because they are charged and therefore polar, they're going to have a hard time moving through a cell membrane, which has this thick, very polar center made of fatty acids. And then there are larger molecules, including glucose, amino acids, proteins that diffuse very slowly, if at all. They may be just too big to diffuse at all. Okay? Now, we've talked a little bit about bacon, and we've talked a little bit about, bacon and its diffusion, and talked a little bit about, stinky potpourri diffusion, and we've talked a little bit about litter box diffusion, but I'd like to talk about a more important physiological case, okay? So let me see if I can just grab, I noticed this morning that okay, so what did I do there? I wanted to grab a couple of these slides and embed them in this PowerPoint. Not there. All right, let's see if I was successful at grabbing those. Sorry, folks. Yay. Okay, good. So now this is embedded here. I knew I would end up having to tweak this, but all right. So what I'd like to do is take a look at this picture. And actually, you know what? I'm going to start at this slide as soon as I get back from a break. I need to run to the restroom. Okay. Can we take a 10-minute break? 10-minute break. So far, the computer's behaving nicely. That's wonderful. I am happy about that. So we are taking an official 10-minute break right now. So get some more caffeine. Probably going to need it, and move around, stretch around a little bit. I got to explain this diagram to you, and because I really want to give you a really good example of diffusion and its importance in cells. Okay, so okay, I'm going to get some more caffeine myself. 10-minute break. All right, Mac. I better check to make sure that see if there's any chat stuff happening that I managed to miss. Uh-oh, where the heck am I? I'm here somewhere. Here I am. Let me just check the chats. Okay. No, I guess not. People are asleep. That's why. Cell membrane transport. It doesn't get any more exciting than that, but I'm gonna turn this over to you pretty soon. This one document that I want you to study and study and study, of course, is one that I'd like to talk about briefly. Now, look, there's a lot here, but let's start up in the top here, okay, where I'm going to show you a drawing that I did. Now take a look. This thing here, this U-shaped thing, this is one of the millions of microscopic air sacs of the lungs, okay? And our lungs have millions of air sacs like this, thin and flat cells on the edge, because thin and flat cells are very useful cells when diffusion is important. And then attached right here is a blood vessel that I've drawn. Actually, it's a capillary of the lung. Capillaries are the smallest microscopic blood vessels. I mean, they're so small that red blood cells, that's what these are red blood cells, have to move through single file. That's how small it is. That means the diameter inside here is probably like 10 or 12 micrometers. Oh, and I have a beautiful model of the capillary that I built myself in my classroom. I wish I could show it to you, but I can't. So notice that the cells that form the walls of this capillary, and it's true, it is cells that form the wall of this capillary. These are also very thin and flat. Now look, if I can, I got to show you over here. Hopefully it won't take me more than a second to find this. There's a document I really would like to show you. That's the one I just opened up, right? And that's where it was not there, okay? So, sorry I'm so disorganized today. I had a lot of this stuff already nicely loaded up and ready to rock and roll, and then it all crashed, when I was looking for stuff, and now, wow, I guess. Hold on. I'm going to try one more time. Taking a folder when I was rebooting everything, taking a folder away and hid it somewhere. Darn. Darn, this was a really cool picture that I somehow managed to lose when I was doing this, and I don't get it. I mean, it's like I said, a file. There was a file up here that seemed to have just disappeared. It was positioned earlier today. Now it's just plain gone. Okay, well, but that doesn't necessarily solve the problem. All right. Whoops. Lots of cell organelles, but darn, every time I think about this image, I think I got to embed this image in here, but it's clearly decided to be as hidden as it possibly can. I apologize. Darn. No, my granddaughter, no, that's not the picture I was looking for. But okay, so I think this means I just need to give up because darn, now I probably find it at some point in time when I'm looking at histology slides. That's where it actually is. Okay. All right. Well, sorry. Um, all right, darn it, I give up. I apologize, wasting all that time. That image was sitting right in front of me, and then I lost it when I booted the computer up. Okay, so let's go back here. I wanted to show you an actual photomicrograph of this because I have a nice photomicrograph. But we're here. Sorry, folks. We're back here. We're looking at this air sac of the lung and this blood vessel attached to the air sac of the lung. Now, here's something interesting. In our atmosphere, when you take a breath, 78% of that breath is actually nitrogen gas. 21% of it is oxygen gas. And less than 1% is CO2. I mean, if you add this all up, 78 plus 21 is 99. All the other gases you might have heard of, like carbon dioxide, carbon monoxide, radon, sulfur oxides, nitrogen oxides, ozone, all of those are in the less than 1% category. CO2, which is one of the gases we're interested in here, is less than 1%. But when you breathe this air into your lungs, it goes deep, deep down into the microscopic air sacs of your lungs. And there it gets mixed with air that's sort of stagnant down in there. And the major effect of it is So from about 21% as it goes in through your nose. And then by the time it gets mixed down in here in the air sac of the lung, it's more like 13% oxygen, right? It's less, it's diluted, so it's about 13%. Carbon dioxide is now way up. It's about 5% in the air sac of your lungs, so big differences. Now, what's important is what's going on in your bloodstream right next to that, because the concentration of oxygen and carbon dioxide in your bloodstream is what's going to drive diffusion under these circumstances. So take a look at oxygen. The average oxygen concentration in your bloodstream, and I had to use an average here because the reality is that it varies a little around your body as to what the oxygen concentration is. So what I had to do is figure out what the actual average concentration was in these deep blood vessels of your body. So look, here it is, 13% oxygen here, 5% oxygen here, 13%, 5%. That is a concentration gradient, right? There's higher concentration of oxygen here than here. That's a concentration gradient. So the question is, can oxygen move through cell membranes and obey the law of diffusion? And the answer, of course, is sure. Of course, oxygen is a molecule that can obey the law of diffusion and diffuse right through cell membranes. So it's 13% here and it's 5%. Well, oxygen is going to obey the law of diffusion and move from the air sac of the lung into the blood vessel of the lung. 13% to 5%. That's what this big blue arrow means here. Where actually oxygen is moving, obeying the law of diffusion and moving into the bloodstream. Why? Because it's obeying the law of diffusion. Now, look over here at carbon dioxide, 6% on the average in the bloodstream, 5% on the average in the air sac of the lung. Well, this isn't as big of a concentration gradient, is it? It's not as big a difference. But the bottom line is that it's still a difference. So 6% here, 5% here means carbon dioxide is going to be diffusing out of the air sac of the lung out of the bloodstream and into the air sac of the lung. This gas exchange that happens right here. Oxygen moves from the air sac of the lung into the bloodstream. Carbon dioxide moves from the bloodstream into the air sac of the lung. This exchange of gases right here is called external respiration. I'm going to write it down here in the chat so you can write it down. Whoops, I lost the chat. What happened there? I'm going to write it down here. This is called external respiration equals, I didn't spell that right. External A and S must be close together. Huh? They are. External respiration is the exchange of gases between the air sac of the lungs and the blood vessels of the lungs. Really, I should say the capillaries of the lungs that are right next to the air sac. And I spelled air sac wrong, so let me go back and correct that so I don't, somebody misspelling it on the quiz or something. So, external respiration, the exchange of gases between the air sac, the lungs, and the blood vessels of the lungs. What's the deal? O2 moves from air sac to capillary, and CO2 moves from capillary sac. The actual gas exchange that happens there. Now, I'll go back to this. That's what we're seeing right here. External respiration. Oxygen leaves the air sac and moves into the bloodstream. Carbon dioxide leaves the bloodstream and moves into the air sac of the lung. Now, I got to ask a question. And believe me, I would love it if somebody would jump in and help me answer it. But look, if this is 13% and this is 5%, then you would think if it just sat here, it'd be 13 and 5, 12 and 6, 11 and 7, 10 and 8, 9, 9, 9, 9, 9, 9. Once it goes to 9, 9, well, then there's no concentration gradient, and you would think this would stop. Right? I mean, there's a certain logic in that. But of course, this never happens until you're dead. Why? That's the question. Why does this never equal out and go from 13 and 5 to 9 and 9? Somebody activated their microphone. Somebody please.
Well, yeah, I mean, the whole law of diffusion is that it never stops moving, right? Or like the Brownian movement, and it never stops. So there's always kinetic energy.
It's true, but the question was, and I think maybe you're on the right track here, is okay, but if you have this gradient 13 and 5 and it moves from 13 to 5, 13 to 5, 13 to 5, eventually you get to a place where you could get to a place where it's even on both sides, and then things would still move around, but you really wouldn't be able to say, oh, it's diffusing in a certain direction. But there was a word that you used that I liked. The word was move. Things are moving here. And that's one of the reasons why it will never even out. What's moving here that will prevent this from ever getting to equilibrium where it's equal in the air sac and in the bloodstream?
Well, your lungs, right?
You bet. Your lungs. What happens with your lungs? I mean,
they're constantly, inflating and deflating and changing the levels.
Exactly. You breathe 14 times a minute. So it means you're basically going to exhale and inhale 14 times a minute. And that means that even though this oxygen concentration may start to drop, it's replenished every 5 times 4. No, it's replenished every 4-ish seconds. Every 4 seconds, it's replenished. And so thanks for saying that. That's absolutely true. And then here's the other thing, of course, is this blood, does it just sit there in a blood vessel? No, blood is constantly moving, right? The heart beats, it pumps the blood into this blood vessel. And does that blood just go and stop? Oh no, this blood is just constantly flowing through. It's going in a loop right here between, in this case, the air sac of the lung and the big toe cell deep down in the big toe. So this will never even out until you're dead, because your blood is constantly flowing. Your heart pumps 70 times a minute, and you're constantly breathing. So you're always refreshing. You're always recreating this difference in concentration all the time. So it will never even out. It will just continue to do the external respiration story. Okay, so thanks for commenting. Now here, this blood flows through here, and once it leaves the capillary of the lung, it goes back into the heart. You know what? I got to say this. I made a mistake here, and it's difficult for me to correct it, but this is wrong. Yay, this is wrong, because it actually went into a capillary, but now it's going to come to a venule and a vein, and then it's going to go back to the heart. Okay, arteries carry blood away from the heart, veins carry blood towards the heart. Okay, so The blood goes back to the heart, then it pumps through the heart and goes into an artery, which goes to arterioles. Like the biggest artery of the heart is the aorta, which is as big as a big garden hose. Blood flows out of the heart and then it goes way down into the big toe. Right? This is blood flow into the big toe. This BTC stands for big toe cell. Okay. Now, honestly, this could be a cell anywhere in the body, but when I think of the big toe cell, I kind of think of a cell that's way out in the boondocks of the body. It has, it is completely and totally dependent on the body's functioning to stay alive and do everything it needs to do. So now we're down here in the big toe, right? So remember the concentration of oxygen and carbon dioxide was an average that I calculated. Well, now we're in a blood vessel. It's a capillary too, a capillary in the big toe that has red blood cells flowing through it. Now they're flowing this way because this is a loop, right? It was in a constant loop. And again, this blood vessel is a capillary, which means it's microscopically small. So red blood cells have to travel through it single file. Now, these cells right next to it, these are big toe cells, whatever kind of cell they actually are. They're deep in the big toe, or deep in the brain, or deep in the heart, or in the tip of the finger. They're somewhere away from the lungs. Now, again, this concentration of oxygen and carbon dioxide, it's the same as here, right? Because this is an average. But let's look inside the cell. Here I am inside of a big toe cell. The question is, what is the oxygen concentration there? And the answer is it's usually less than 1%. So we see a nice pattern here, right? 5%, less than 1%, 5%, less than 1%. Now, at the same time in the big toe, the carbon dioxide concentration is very high. 8% is dangerously high, like poisonously high. And 8% in the cell, 6% in the bloodstream. We, again, absolutely see a pattern here, right? The bottom line is that if it's 8% here and it's 6% here, it's going to move along this concentration gradient, down this concentration gradient, and it's going to move, carbon dioxide is going to move from where it is inside the cell into the bloodstream nearby. Okay. At the same time, oxygen, you notice, is going to be 5% inside the blood vessel and only 1% or even less than 1% inside the cell. So we can see what's going to happen there, blue arrow, of course. 5%, 1%, oxygen is going to move out of the bloodstream, in this case, this capillary, and into the big toe. So we see a pretty clear pattern here too. Oxygen diffuses out of the capillary into the big toe. Carbon dioxide diffuses from the big toe into the capillary. There is a name for this that I want to write down here. This is called internal respiration. And internal respiration means I'm just going to copy this for a minute, paste it in here, and hopefully correct it. Okay, because this is internal respiration. All right, let me just correct it. The exchange of gases between the blood vessels and the deep internal body cells oxygen moves, in this particular case, oxygen moves from the capillary I'm going to post this any second. All right, I hope I didn't screw this up. I should just type it out and not. So let me just quickly proof it before I post it. Internal respiration, the exchange of gases between the blood vessels and the deep internal body cells, such as big toe cells. Oxygen moves from the capillary into the big toe cell, and carbon dioxide moves from the big toe cell into the capillary. I think it's right, okay? But there it is for your note-taking purposes. Internal respiration, external respiration. Now, again, you might look at this and think, well, what do you mean by internal, external? Well, look, the air sac of the lung is pretty darn close to the outside world. This is a respiration. It happens pretty close to the outside world, hence external. This is an exchange of gases that happens deep inside the tissues of the body, like the big toe cell or some other deep tissue cell. Now, does anybody have any questions about this internal respiration, external respiration? Some of it's repeated in the PowerPoint after this, but I wanted to get some good definitions right there for you, okay? I was hoping for a question, but here's the thing. Why, I can't resist this, why is it that the oxygen concentration inside these cells are so low and the carbon dioxide concentration is so high? I mean, why is that?
Is it because the cells like absorb the oxygen?
Absolutely, the cell absorbs
the carbon dioxide.
It's taking it up. It's picking the oxygen up, and that's part of the story, absolutely. But the rest of the story has to do with this process called cellular respiration, and it's cut off at the bottom. I'm sorry about this, but I've already talked a little bit about there. This process is called cellular respiration, and this is the process where a cell takes food molecules and breaks them apart very carefully to liberate energy. So cellular respiration, the biochemical pathway, describes how food molecules are carefully taken apart to extract the energy from and store in ATP. And honestly, it looks like this in a very basic way. It looks like, let me just write this on a different piece of paper so I can import it here. It will just take a second, but I think it'll be easier for me to write it and use it if it's already pre-written. It won't take very long. when I rebooted, when my computer was updated and rebooted, it just plain messed with everything. Because here's what I want to write. I want to write this. C6 H12O6. Well, uh-oh. O6 plus, this is a chemical reaction, oxygen gas so much easier when you have a board to write on. You can pre-anticipate that. Okay, see if I can make this look right. Absolutely should have enlarged this. It would have probably made it a tiny bit easier to deal with. All right, so here's what I wanted to paste in the chat line here. Find the darn chat line again. Well, I'm going to try and get my act together a little bit better tomorrow, folks. I don't know why. Just feel like today those computer problems really set me back. But there it is. Oh, gosh, after all that work, it didn't keep the formatting anyway, right? So this is what the equation looks like. I'm sorry, it didn't. It's C6H12O6 plus O2 reacts to form CO2 plus water. But why did we do it all? To get energy out and stored in ATP. Now, that's exactly what it says here at the bottom of this. It says cellular respiration, sugar plus oxygen reacts to form carbon dioxide plus water plus energy. So here's the thing. Notice that on the left side of this equation, there are two reactants, sugar, which means glucose in this case, C6H12O6, and oxygen. And when these two are together in a cell, the cell is going to rip this sugar apart using this oxygen, produce carbon dioxide gas, plus water, plus energy that it will store in ATP. But here's the main point. The original question was, why is oxygen concentration in cells so low? And the answer is because oxygen is a reactant in a super important chemical reaction. And that means that as soon as oxygen moves into a cell, it gets incorporated into this reaction and it gets used up. Now, the oxygen's not destroyed. The oxygen ends up over here in this water. But it no longer is oxygen gas, and so that explains why the oxygen concentration is so low. Now, why is the CO2 so high? Well, again, take a look at this equation. the products of this reaction, which liberate energy, are carbon dioxide and water. So in this cell, what's happening is oxygen and glucose are going into the cell. They're being metabolized. And what's going to be left is carbon dioxide. That's the leftovers from the sugar that you broke down, plus water. But water is always an issue for cells. So cellular respiration explains some of the underlying chemistry for why these diffusion gradients exist. And I remember many years ago reading a respiratory therapy textbook, and they talked about something called the respiration triad. And the respiration triad internal respiration plus external respiration plus cellular respiration equals the respiration triad. And this book actually recommended that you use this acronym here, ICE, to remember it, which doesn't stand for what? Immigration Customs Enforcement. It stands for internal respiration, cellular respiration, external respiration. These three respirations that are super important. Again, I'm going to find that image and show it to you later because I would want to embed that image in here somewhere so that you could see an actual photo micrograph of exactly what this looks like. But the bottom line is that why I talk about this at all, and I've spent too much time talking about it, is because, look, I talked about bacon diffusing, I talked about potpourri diffusing, I talked about stinky litter box smell diffusing, but this is a real physiological thing. I mean, people kind of know that you breathe in oxygen into your lungs, and it goes into your bloodstream, and then it gets pumped around to your organs and other structures of the body, and then it moves out of those blood vessels into your cells, so that your cells have enough oxygen and they can get rid of carbon dioxide and all that. But most people don't really know in any kind of real picture about exactly what that looks like and why that's so physiologically important. So now you do know why it is so physiologically important. All right, any questions on ICE and the respiration triad? Okay, respiration, three functions. Organisms supply oxygen to their cells. Cells use oxygen to produce high energy molecules, that's ATP, and carbon dioxide waste is removed. Whoops, sorry. It happens in three stages: external respiration, internal respiration, cellular respiration. External respiration is the exchange of gases between the alveoli, that's the microscopic air sacs of the lungs, and the blood vessels right next to and attached to the alveoli. Internal respiration is the exchange of oxygen and carbon dioxide between blood and cells. So the oxygen goes from a lung cell into the blood, into the body cell, and then carbon dioxide goes from the body cell into the blood and back into the lung cell. You've got both of those going on, right? So let's just make that clear. Oxygen moves from the air sac of the lung cell into the bloodstream and ultimately into a body cell, okay? I'm gonna make these smaller. moves from carbon dioxide moves into the body. Carbon dioxide moves into the bloodstream. Let's go. Into the bloodstream. Well, that's a new word, at least. Okay, almost there. Wanted to try and make this as oh, that wasn't right. All right, there we go. So oxygen moves from the air sac of the lung into the bloodstream, then from the bloodstream into the, let's just say, use the same terminology, deep body cell. Carbon dioxide in the deep body cell moves into the bloodstream and therefore into the air sac of the lung and moves from the into the outside cell. Oh, okay, I better save that so that you have it. I'm going to post, of course, I'm going to post this document as soon as we're done today. So there's images here that depict this pretty well. And there's cellular respiration, which I already mentioned, the chemical process within cells that takes glucose energy. It takes glucose, it tears it apart. Where was that? Let's see if I can grab that off the. I'll grab it off of there, Steve. All right, good. That looks good. Cellular respiration. All right, so there's the equation. C6H12O6 plus O2. Big biochemical pathway involving mitochondria produces CO2 and H2O and energy, of course. So, anyway, ice, the three stages of the cellular respiration, all of this is absolutely distinctly related to diffusion. Okay, and a very concrete example of diffusion. Now, let's talk about osmosis. Let me just, for 1 second, go down here. I just noticed that maybe it's up here and I somehow missed it. I guess I just mentioned carrier-facilitated diffusion very quickly here, which is the type of diffusion in which a carrier protein is used to move things across a membrane. So I am going to put a specific example here that I'd like you to be aware of. So one of the molecules that is moved into cells by carrier-facilitated diffusion is, of course, one of the most famous and important molecules that cells absolutely have got to have. And the concentration of glucose is always higher outside of cells than inside the cell. And the reason is pretty straightforward. The minute that glucose gets into a cell, it gets torn apart in cellular respiration. So we've mainly been talking about simple diffusion, but carrier-facilitated diffusion means that you have a protein carrier that's embedded in the membrane. It is a pore that is designed for the passage of molecules. It can be general or selective, but the most famous one is the glucose transporter protein. Usually glucose is about, whoops, so the glucose concentration outside of cells is pretty high. The glucose concentration inside of cells is pretty low, and that is just because the minute that glucose gets moved into a cell, it gets torn apart to form. Well, it's torn apart in cellular respiration, no doubt about it. Okay, again, I'm not sure what's going on here, but Okay, so just for the record, big difference in concentration. You would expect glucose would move into cells. The problem is it's too darn big, and it won't move into cells in a way that allows cells to acquire glucose as fast as they need to. So modify that so that we are talking about carrier facilitated diffusion. So the word carrier refers to carrier protein. Facilitated means half. Excuse me, half. Facilitated means helped. It helped the diffusion happen. And then, of course, it's diffusion, which means it's obeying the law of diffusion, moving from where it is more concentrated to where it's less concentrated. And honestly, I don't know many examples of carrier-facilitated diffusion, but one of the most famous, of course, is good old glucose. And it is moved by concentration gradients. Okay, I'm back down to here. All right, so now we can talk about osmosis. And when I think of osmosis, I sort of think of gummy bears. I shouldn't really, but I do think of gummy bears. Let's talk about osmosis. Most processes in our bodies and in cells take place in water. Most cells, 70, 85% water. I mean, this is from a purely molecular standpoint. There's somewhere between 70 and 85% of the molecules inside of cells are water. Water molecules are small and they diffuse easily across membranes through small pores. You remember what these pores were called? Well, like I said, I'm glad I didn't post this a priori. It's much better that that I'm able to make these tweaks, and when I post it, all the tweaks will be there. Okay. The diffusion of water across a membrane is called osmosis. So, how is osmosis different than normal diffusion? Well, number one, it is only water. It only involves water. Never anything else. We're only talking about the movement of water through a membrane. And of course, that membrane is usually a cell membrane. I mean, that's the kind of thing we're thinking about. In this particular case, there's water molecules here. These red things are not water molecules. Those, as you can see down there, are solute molecules. This is stuff dissolved in water. So over here, the water is actually pretty highly concentrated. Over here, it's less concentrated. Water is going to move this way and obey the law of osmosis.
I'm sorry,
So,
could you repeat the definition of osmosis? Did you say it's the diffusion of molecules across water?
I would be happy to. Thank you. Let's go back and let's do it this way because diffusion of water across the membrane is osmosis.
Cool.
Yeah, but hold on. I'm gonna say this. Osmosis. I'm gonna erase that. Only water Let me correct this. Now, what is worth making a mental note of is whenever anything moves from where it's more concentrated to where it's less concentrated, we call that diffusion. If you're talking about a scenario where the molecule that you're interested in is water, and there is a cell membrane evolved, that is a kind of diffusion, right? And so you can define this way. Osmosis is the diffusion of water across a membrane, right? And diffusion implies concentration differences, right? Implies it. Now, I've just redefined osmosis here again. Osmosis is the movement of water through a cell membrane from where water is more concentrated to where water is less concentrated. Right, so let me save that. Make sure it's up to date and all that. Go back here to the slideshow. Nope. Cancel. So did that help? I put a different definition for osmosis. Oh, and I guess I didn't. I made it too big.
Yeah.
I'm gonna get it. I thought it would all somehow squeeze in there when I did that, but I guess not. I'm gonna make it all available. It's gonna show up. Oh, anyway, thanks. I think you were saying, yeah, Steve, it made a difference.
Yeah, it's yeah.
Okay, good. That's what I wanted to do.
Also, so earlier when I was just doing some review, I looked up the definition of concentration, and it's I got the amount of substance in a defined space as one of those. Is that accurate, more or less?
It is accurate, more or less. Okay. Meaning that if you're just asking, look, what does the word concentration mean? It means kind of how much of something there is in a particular place.
Okay, yeah. Perfect.
So you can talk about all kinds of things when it comes to osmosis, but what's critical that we need to think about is what's called water concentration. Okay, it's the water concentration. Okay, so I'm going to just go. I'm not going to try and put it on that last one. I'm going to put it on here. Now, I'm sad to say you don't have Goldenrod Packet page 23. That was something I used to give out in my classes, and I don't have a clue how to do that now unless I want to sell it or something, right? But it's a packet I used to give out that would, I think, be very useful. Now I'm just going to change it. So when we think about osmosis, we always have to think about water concentration, because that's what we're interested in, is water concentration. Now, look, I got to tell you, when it comes to osmosis, I personally probably have sat through 10 different lectures on osmosis, and most of the time after after the lecture was over, if you asked me, well, do you understand what osmosis means, Steve? I would have said, yeah, sure, I understand it. But as soon as you queried me in any detail about it, I would realize that I'm not sure I totally understood it. So I have to tell you that eventually a professor pointed out something to me that I think is important to understanding osmotic patterns and things, and that is this. It has to do with a solution. What is a solution? And a solution, you may remember this from the McDast stuff, is a solute dissolved in a liquid solution. The solute is what's dissolved, like salt in seawater. Okay, salt in seawater. Seawater is about 3.5% salt. Plus, if it's 3.5% salt, then it's 96.5% water, right? The solute is what's dissolved in the water. And we're talking about biological systems then. We're talking about water. A solvent is what the solute's dissolved in. I mean, in biological systems, that's water. You think about water solutions. I mean, okay, you could go to the liquor store and buy a bottle of 151 proof rum, and 151 proof rum is 75.5% alcohol and 24.5% water and coloring and that kind of stuff. So obviously that's got a lot of alcohol in it. But the bottom line is that most of the time when we think about solutions in a biological sense, we're thinking about water solutions. Okay? And the tonicity is sometimes called the strength of the solution or the concentration of the solute. Now, to understand that there's this inverse relationship, okay, that inverse relationship is important and definitely worth putting in here. Okay, so here's how I'm going to phrase it. As, remember the hard brackets? A concentration of, as the solute concentration increases, right, to squeeze all of this in, I need to just do a little bit of playing with this. So what I'm just trying to illustrate here is the relationship between solute and solvent. And what I mean is that when you look at solutions, there's two things to consider. There's the dissolved stuff and its concentration, and then there's the water concentration. And when we talk about osmosis, we mostly want to think about water concentration, since solutes usually don't move, but water will move through a cell membrane. So notice a 10% salt solution is 90% water. 30% salt solution is 70% water. A 60% salt solution is 40% water. A 50% salt solution is 50% water. They too have to add up to 100% of the solution. So just some concrete examples of this. They're inversely related. As one goes up, the other goes down. So if the water concentration is water concentration goes up, the solute concentration has to go down. You don't really have any choice. It must go down. Okay, so I personally found that when I tried to understand this, it made the entire idea of osmosis easier to understand. Now, this is kind of an interesting thing. Osmosis can create a pressure that has to do with the movement of water through the membrane. So let's just say we start here where we've got not much solute, and over here we've got lots of solute, okay? These dots represent solute. So actually the water, if the solute concentration here is less than here, then that means the water concentration here is greater than here. Remember, they're always related backwards. If the solute concentration here is less than here, then the water concentration here must be greater than here. So what's going to happen is the water is actually going to flow through this membrane, and it's going to sort of dilute what's going on here, which is going to make that column climb up and this column drive down. And the pressure that occurs as a result of this, the osmotic pressure, is a real measure of how water wants to move and will move through a membrane. Now, in reverse osmosis, which you've maybe heard of, where they use it to purify water, they actually put a pressure that pushes water molecules from where they're more concentrated to where they're less concentrated, and therefore it pushes water molecules through a membrane and creates purified water. So when it comes down to these, this tonicity, here's the big three. These are the three possible scenarios, three possible scenarios, okay? One scenario is called the isotonic scenario. The concentration of the intracellular water, I better make sure that this terminology is clear enough here, okay? So let's just go down here a little, and I'm going to put up here EC equals cellular water I can spell. So just for the reference, we've got these set up here. So in case you're wondering what IC and what IC and EC stand for, there we go. Isotonic. In an isotonic solution, the concentration of the intracellular water is the same as the extracellular water. In a hypertonic solution, the concentration of the intracellular water is higher than the extracellular water. And in a hypotonic solution, the concentration of so in a hypotonic solution, the concentration of the intracellular water is lower than the extracellular water. So here, really, got to reverse this here, that's all. Okay, so some quick abbreviations here, ECI, etc. It might make this a little bit more understandable. I don't know. We'll see. We're still talking about a beaker with a cell. You know, I remember that this happened to me last semester, and I didn't realize at the time that things were backwards, but I didn't really care at the time because when I draw this, what I'm drawing is some kind of beaker like this. Okay, here's a beaker, and inside the beaker, here's a cell. And it doesn't make any difference that this is, uh-oh, it might make a difference that this is going to be reversed. But look, here's my picture. Here is, it doesn't make a difference. The exclamation, the question marks are backwards, aren't they? I know how to fix this.
Regular video that's up, it's normal. So I mean, I can see both your presentation and your regular video, so I can see it normally.
But doesn't this look, I mean, is the question marks backwards on this?
On the presentation it is, yeah. But
on this. Yeah, on my silly drawing.
Huh?
On my drawing, it's backwards.
Yeah, right? But it's only backwards for one view because I have two views. I have a view of like just.
Oh, that's right.
And then the view of you in the presentation.
Okay, but again, so it's one. Now, here's what I can do though, is I know that one thing I can do is I can stop presenting for a minute. Then I can show this. Can people see it? No, because I'm not
Yeah.
presenting.
Yeah,
Nobody can see this.
I
Or
can see
people,
it.
you can see it. And does the question
It
marks
looks
look
normal.
now? Okay, good. And then I'm going to write next to it this, which should look normal for now. Okay, and so I'm kind of picturing this cell that just got dropped in a beaker full of some solution, right? And so there's the cell dropped in a beaker of solution. And there's a question mark inside the cell. And there's a question mark outside the cell. And the question marks, the question sort of is, what do the question marks refer to? And here's what the question marks refer to: water concentration. If the water concentration inside the cell is greater than outside the cell, then guess what's going to happen? Water is going to move from where it's more concentrated to where it's less concentrated. And as a result, the cell is going to shrink and lose weight. It's going to lose weight because water weighs 1 gram per mil. Now, what if the water concentration outside the cell is greater than inside the cell? Well, then water is going to move from where it's more concentrated to where it's less concentrated. And water is going to move into the cell, and the cell is going to swell up, and the cell is going to gain weight. Because, after all, water weighs 1 gram per mil. Now, the question is, what if the water concentration is equal on both sides? Well, if it's equal, then certainly some water molecules are going to move out and some water molecules are going to go in. But you're not going to see this big difference between a bunch of water leaving, a bunch of water entering. You're basically going to see nothing. If a cell is dropped into a hypotonic solution, the hypotonic solution, high water concentration, water is going to move into the cell's going to swell up and burst. Oh, good. Especially if it's a blood cell. Blood cells are the most. If you drop it in a hypertonic solution so that the water concentration inside the cell is greater than outside, it's going to move out. The cell's going to shrink and shrivel up and lose weight. But if it's exactly identical, 99% here, 99% water here, 99% water here, 99% water here, 99% water here, then really you're not going to see any pattern of movement. And I need to figure out how to draw that and put that on a PowerPoint, but so far I have not figured that out. Okay. What did I just do? Did not want to do. I wanted to go back to the good old slideshow. All right, so remember what I said a minute ago? Isotonic means equal concentration. Hypertonic means the intracellular water higher than extracellular water, so the cell's going to shrink and all that. Sorry, hypotonic means that the cell has found itself surrounded by a solution that is more concentrated water than inside the cell. And that's going to affect the cell. Like if the cell it has a low solute concentration, then it has a high water concentration and water is going to leave. If the internal cell has a high solute concentration and a low water concentration, then water is going to enter. And the cells we mostly need to worry about in this case are not like a lung cell or a blood or a big toe cell. It's a blood cell, because blood cells are free-floating cells. They require themselves to have a certain shape in order to function right. And so what's going to happen is if you float a red blood cell in an isotonic solution, equal water inside and outside, that cell is going to stay, it's going to be in what is sometimes called a normal solution. I'm going to go back here. Just write that down here. Isotonic. Come on, don't be a troublemaker here. Isotonic. This is also referred to as a normal solution, because if you drop a cell into it, the cell is going to stay normal. It's not going to swell up and gain weight. It's not going to shrink and lose weight. It's going to stay normal.
You're not presenting anymore.
Oh, thank you. Thank you. Thanks for reminding me. I made myself a mental note a few minutes ago to say, remember, you're not presenting, and so you better go back to presenting if you start trying to present anything. And nevertheless, I forgot it. So thanks so much for reminding me. That's easy enough to correct, I think. I don't anticipate doing anything different than where I need to go, but frontwards, backwards. That's just too much, too crazy. Okay. So back here, that's what this normal means. An isotonic solution is a normal solution, and a normal solution is, a normal solution. I'm just curious as to whether I can. I'm going to fix this. Don't worry. Take me a second to fix this. Whoops. So this first scenario right here, where the cell remains normal, remember, that's called a normal solution, and the cell stays normal. But over here, if you drop a red blood cell, especially in a hypotonic solution, what's going to happen possibly is something called hemolysis, right? And then if you drop it in a hypertonic scenario, it's going to be called crenation. I got to get rid of some of this, and I will reposition everything just right. And I think this just take a second. I want you to be familiar with these terms. There we go. So one possibility is that the cell will actually swell up and and explode. I mean, red blood cells are absolutely the ones that are most likely to have this happen. White blood cells could be the same thing. Hemolysis, what a great term, right? Hemolysis means for blood um, to explode and be lysed. Lyse means to split apart. Now, if instead it shrivels up like this, it is called crenation. And this is when you drop a cell into a scenario where the water concentration outside the cell is lower than inside the cell. So what does water do? It has to obey the law of osmosis. In a plant cell, you might actually see the cell membrane pull away from the cell wall. But in a blood cell, like a human blood cell, it would simply shrivel up. Now, it wouldn't be destroyed. You might be able to rehydrate it and use it again. But once a cell like this is exploded and lysed, well, then it's it's impossible to really reclaim it. I had a student many years ago who told me about, it was an EMT, and told me that if you find somebody if you get a call and you have to save somebody who has drowned in a freshwater lake, then you have to consider that they have been exposed to a hypotonic solution. And that hypotonic solution is means that the cells of their lungs are going to be exposed to water concentrations higher than their blood cells, for sure, and therefore they could in fact have blood cell damage as a result. And so, of course, what you're going to do is you're going to get the water out of their lungs as fast as it can. Then you're going to get them breathing and all that. But when you're taking them to the hospital, you might consider actually giving them a blood transfusion, because if they've been exposed to a hypotonic solution like this, then their red blood cells and white blood cells may have been seriously damaged. I mean, after all, think about it. You drown in a freshwater lake, you're bringing fresh water, which is hypotonic, into your lungs, way deep down into the air sacs of your lungs where it gets exposed to your bloodstream. And that water is going to flood into your bloodstream, and it could ultimately cause hemolysis, or blood cell destruction. So if you're in a position where you can give that person a transfusion on the way to the hospital, that would be great. Now, the scenario is slightly different for salt water, because salt water is a hypertonic solution. So when you breathe that into your lungs, you're now exposing your bloodstream to a hypertonic solution. And a hypertonic solution means it is going to suck water out of your bloodstream, and it's going to make your red blood cells undergo crenation, basically. So under those circumstances, he said, okay, we got to get the water out of the lungs. We got to get their heart started. But then on the way to the hospital, if we have it, we want to try and rehydrate their blood, because their blood cells may be all created. We want to rehydrate their blood and get fluids into them. So they might give them a normal glucose solution that just rehydrates their bloodstream. And honestly, a normal glucose solution or a normal saline solution means that what you're putting into the bloodstream is a solution that will rehydrate the body. So it is worth mentioning, and I'm gonna scan this down a tiny bit. It's worth mentioning over here under normal. Do it. If I can do it. Drink this a little bit more. Because under normal, what I'd like to put is this. 0.9% solute. Because that's actually the normal value. Right? And then I can put it, that means if it's 0.9% solute, it means it's 99.1% water. All right, I'm not sure what this is going to look like, but we'll see. Oh, not good. Okay. I move everything down a little bit. That's all. I think that's totally doable. Yay. Okay, good. Oh, all right. So, normal solution. I don't know if anybody remembers, but I said this before. Salt solution in the ocean, 3.5% salt, meaning 3.5% solute. That means it's 96.5% water. And so it is actually a hypertonic solution. Okay. So I don't know. I almost wanna just mention here, if I can go over here, it's crazy. What kind of solution? Get it. Distilled ether? No. Water? Tap or distilled water, even pond water, would be a hypotonic solution. Okay, and then what about crenation? What would cause that? Well, I mean, I said this already. Ocean water is a hypertonic scenario. When I was a kid, my mom, would always, anytime, if I dared to say I had a sore throat, my mom would immediately sit me down and make me gargle with a warm salt water solution. And that warm salt water solution, it was disgusting. It was absolutely hypertonic. and it it flooded the cells of my throat with a hypertonic solution, which you might guess would then make them would make the cells of my throat. Let's see. That we're going to put over here. Would make the cells of my throat shrink, and all of a sudden my sore throat would go away. Now, in my house, I honestly have to say I cannot think of anything that's a normal solution. Well, I've got some bags of normal saline that I use to hydrate a cat with kidney disease, so I guess I've got that. But beyond that, I really, only thing I can think of in my house that might have, wow. I'm really pushing my luck here. I wanted to give you some more details. When we're done with this, I'm gonna turn the rest of the PowerPoint over to you to look at. All right, now this looks kind of crazy to me. Let's just, out of pure curiosity, I gotta take a look at what this looks like. No, I think there's a slight possibility that it might work, so we'll see. Tons of data packed into this, and so let's see. Nope, isn't there, huh? Where the heck did that go? I do not know, huh? Well, I guess better. Wow. Moving this around like that. Hypotonic, hypotonic, isotonic, hypertonic. Okay. I really messed that up. Just trying to figure out where this stuff is. I think it's just buried somewhere. Okay. That's where, all right, so here we go. Hemolysis. Filled. Like I said, my mom would always make me do this saltwater gargle, which was disgusting, and I thought I was going to throw up the whole time, but it would cause the cells in the lining of my throat to undergo crenation. And once they underwent crenation, they would shrink, and that would make the entire, I mean, that would make my throat hurt less, and that was a good thing. Well, I think I've got it down there, and that includes actually some answers to, I mean, there's some stuff in here that's embedded here that includes some answers that could be applicable to the upcoming quiz. What I wanted to put over here for normal, and it somehow I couldn't get it to squeeze in, even though I thought I should be able to get it squeezed in, was the only example of a normal solution that I would actually have in my home would be some ancient contact lens solution that I haven't used in a long time. Right, and I feel like that should be there. Let's see. And that contact lens solution says right on the bottle, normal saline. All right, normal saline. All right, things are fine here. I need to absolutely escape from this. Oh, it's crazy. Something's going on behind there. Who knows if I can ever fix that? I'm not going to try it. All right, so look, here's the thing. I'm going to stop on this PowerPoint right now. This has been a crazy day. I apologize. Lost a bunch of time. Spent too much time talking about osmotic scenarios. However, if you're going to understand the whole gummy bear story and what's happening, you are going to need to understand this basic osmotic stuff. And so I wanted to cover it. Now, look, there is a bunch more in this PowerPoint, and I am leaving that to you, okay? So you go ahead and look at this stuff, my advice might be that if you have the time and energy to do it, you come to something like phagocytosis, which is a active membrane transport process. Well, gosh, I got to tell you, when it comes to phagocytosis, hey computer, I would be so tempted to go to YouTube, just write in, oh, I don't know, just phagocytosis. And here's a really famous phagocytosis video. It's actually a video of white blood cells chasing and engulfing bacteria.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
I don't know if it really makes a noise like that, but I'm just saying that there's great videos of many of these cell membrane transport processes for sure. And while you're going through this PowerPoint, which I'm going to post this PowerPoint as soon as we're done with class in a few minutes, okay? So it will absolutely happen in a few minutes. I am also, when I post this PowerPoint, I am also going to post for your perusal. I'm going to post this PowerPoint on DNA and RNA and protein synthesis, and I'm going to ask you in your copious free time if you would take a look at it, because tomorrow we're going to boom right through it. I'm not kidding. We're going to boom right through it. I know it. We're going to boom right through it. Okay, so I'm going to post this one too. It's called Cytology 4 DNA and RNA and protein synthesis. Okay, that's going to get posted also. Okay. All right, now back to class, I guess, huh? Where am I? I'm somewhere down here. Well, look, I apologize. Today was just plain crazy. Now, I did pack a bunch of information into that osmosis thing. Certainly in the quiz that I'm getting ready to post tomorrow, I'm going to ask questions about cell membrane transport. And since this quiz is going to extend over, I don't expect you're going to see it tomorrow morning and you have all day Tuesday and all day Wednesday to work on it. Of course, it's going to include questions that come from Tuesday's material and Wednesday's material. Okay, so don't just to be clear about that. I hope I was presenting the rest of the time. I am presenting now. So we got the neutrophil chasing bacteria embedded in here too. It's part of a cell membrane transport process. Any questions? Is anybody awake? This is designed to wake people up. And I have not told a single biology joke in I don't know how long. I used to think of biology jokes all the time, but I'm going to give myself a round of applause anyway. Man, I'm telling you, my butt really hurts. I mean, we've been working way too hard today. Tomorrow, it's going to be just as bad. I have no doubt about it. Okay. So, bottom line is that we're done for today. Lots of stuff to think about. I will get that quiz posted ASAP. If I can get it posted tonight, I'll get it posted tonight. Otherwise, it'll be posted tomorrow morning. Right? Thank you so much for coming to class. I really appreciate it. This is the last full week of classes, and somehow I'm going to get through as much material as I can get through, and then that will be what we get through, I guess, right? All right, so don't forget, please look at those 2 PowerPoints. I'm going to post those PowerPoints as soon as we are done, meaning as soon as I say peace, stay safe, avoid that darn virus at all costs, and I will see you tomorrow. Okay? Hang in there,
Thank
folks.
you.
I'll see you tomorrow.